my voice okay, ma'am? Okay, can? Can,
Good morning, Bell.
Alright, so question 4 is where we are going to continue. Alright, so in genetics, like I said already, you have to read the question carefully and get some clues. Right, so you have two parents. So the first parent is a homozygous white flowered long stem tobacco plant. The second parent is homozygous pink flowered short stem plant. Alright, so over here. Okay, so besides homozygous, sometimes they can use other terminologies. So what other terms can they use besides homozygous? They can say pure breeding. They can say pure breeding as well. Okay, or they can say pure line. Okay, all of these terms refer to homozygous. I mean, it's the same. Alright, now after that, they say that the resultant progeny have white flowers and long stem, and this is basically the F1 plants, and the F1 plants, they were self, okay, to give you the F2 generation, and you see 4 different phenotypes. So the first thing that you need to do is to decide, okay, for your genetic diagram, right, where should you start from? Are you going to start from F1 generation to show you the F2? Or are you going to start from the parental generation, then the F1, then the F2? I think this is a question that a lot of students like to ask. Do you need to show the parents? Yes, okay. How do you know that you need to show the parents? Really, it's dependent on the marks. Right? You look at a lot of genetics questions, generally it's 4 marks. But this question is 6 marks. So you have to show the parental generation. Okay? So we start off with a symbol. Alright, the symbol, there are 2 gene loci. So this is the 2 gene loci. So the first gene loci is for flower color. The second gene loci is gene B for height of stem. Alright, 2 different gene loci for 2 different characteristics. Okay, now how do you know which is the dominant allele A? Is it the white flowers? Is it the white flowers? Is it the allele for white flowers, or is it the allele for pink flowers?
Why?
How do you know?
F1.
Very good. You're going to look at the F1 generation, right? So if you cross 2 different phenotype parents, but you see the F1 generation only having white flowers, it would mean that, okay, white flowers is the dominant trait. Okay, then if you look at gene B, which is the height of stem, right? Which one is the dominant allele? Okay, it's the allele for the long stem. Okay, so probably the enzyme code for, I don't know, I mean the allele code for an enzyme that results in stem lengthening or something. Okay, all these genes, right, is talking about a certain type of protein that is made, either an enzyme or something. Okay, whereas just now that allele A, big A, is probably an allele that codes for enzymes that leads to production of maybe no pigment, so it's white in color. Alright, anyway, so we will look at the F1 trait to see which is the dominant trait. Alright, so the long stem is the dominant trait. So first thing, what should you do when you're doing the genetic diagram? You are supposed to write the key. Okay, so please write the key. Okay, so after you have the key, which is your symbol, basically, you're gonna continue. Okay, so in your key, alright, you tell me what alphabets you're using. Okay, I recommend you use 2 different alphabets that is quite different between the capital and the small letter. Right? Sometimes students, right, they like to use things like Y. It's not very clear. They look quite alike. Or they like to use W. It's also not that clear. Okay, so something that is very different between the big and the small letter will be good. Alright? Okay, so anyway, after this, you have to write the parental phenotype, so that one shouldn't be a problem, and then the parental genotype. Okay, now the gametes is a problem for many students, okay, but I think by this time, I hope you all don't have an issue anymore. Alright, so over here, okay, parental phenotype. Okay, I just want you to point out, right? You're supposed to write white flowers, long stem, and you need to write a cross in between because you're crossing 2 different parents. Okay, Now, in this question, did they say the gender of the parent? Did they say like male part from this plant, female part? No. So there's no need to write the gender. But in other questions, if gender is specified, please put the gender as well, especially for sex-linked questions. Okay. Now, parental genotype, no issue, I think. Okay, because they are homozygous, so it should be all the same alleles. And the gametes, okay. Now, common mistake made by students. Some students are gonna write this. This is the most common mistake. Now, when you look at your own answers, your gametes shouldn't be like this, right? Every gamete, you should have one allele of each gene. Okay, so for gene A, you're gonna get one of the two alleles, and gene B, you're gonna get one of the two alleles. This is Mendel's law of segregation. Alright, so make sure you don't have that. And after that, you get the F1 genotype, no issue, right? So you just do a fusion, right? And your F1 phenotypic ratio will be this. Okay, so what are the marks awarded to for genetic diagram? Okay, so this is a common question asked by student. So usually, the genotype and the phenotype of the parent is 1 mark. Okay, for this case, I think because the phenotype is given already, so the genotype is 1 mark. Alright, so the gametes, okay, it is another mark, and make sure you circle the gametes. The moment any gametes is not circled, no marks. And lastly, the F1 phenotype, okay, is also 1 mark. F1 genotype and phenotype. Alright, yes. Oh, don't need to draw arrows. That one secondary school. Any other questions you have? By the way, I don't know if you all realize, right? For ratio, right, it need not always be 1:1, 3:1. Even when you say all is considered a ratio, because I mean all plants are white, right? All of them are white. Anyway, after that you do a self. So self means what?
Own self.
Own self. Okay, so in plants it's possible to self. So because plants have is it called an anther?
Yes.
Anther, right, that has like pollen grains, and then after that you have a female part called a stigma, right? So you can just self that own plant, right, by transferring pollen grain from the what is it? Anther to the stigma. Okay? So anyway, if you were to self-pollinate, right, your F1 phenotype, your F1 generation, these are the gametes that you should have. This shouldn't be difficult, you have done it in lecture already. So basically, in the gametes, there's 4 different possible gametes. Yes. Oh yeah, is there a typo? Oh yes, there's a typo. Wow. Okay, so there is a typo. Alright, so basically, why are there 4 possible gametes formed? Due to Mendel's law of independent assortment and segregation. Okay, remember? So because your homologous chromosomes can arrange in different ways on the metaphase plate, right, you're going to have 4 different types of gametes formed because the alleles of each gene can combine with each other in different ways. Okay, so I hope you know already. And based on this, okay, you have to draw a Punnett square to show the F2 genotype and phenotype. So another question asked by student is, wow, must I write this inside here? Very leceh, you know? All this like white long, pink long, do I need to write? Yes, you have to write in your Punnett square, you'll be awarded marks for it. Because right now this Punnett square shows not just the genotype but also the phenotype. So with every line you have to describe it. Okay? Yes, all that for 1 mark. Right, if you are desperate, right, if you are really, really desperate, okay, like let's say you only have 1 minute left in your exam, right, you can try your luck, you can write white, okay, and then you write, oh, okay, it means W, and then long means L, and then after that you can just write white, long, whatever, you can write a key lah, basically. Okay, if you are really out of time, but if not, please spell it out. Okay? Any questions? Oh, another question asked by student, this one. Is it compulsory to write this? Okay, you all know what the symbols mean, right? Okay, male and female. So for Punnett square, usually by convention, we'll just write it in. Okay, but we will only mark for it, like double check that the gametes are from the male and then the gametes are from the female if the gender is mentioned in the question. Otherwise, you can just put randomly. Any other questions you have? So a common mistake by students is this. For some reason, I don't know why, okay, but when they draw the gametes inside the Punnett square, they always forget to circle the gametes in the Punnett square. They will remember on top, but in the Punnett square, they'll forget. So make sure you do that. Alright, then after which you get all the genotype and phenotype. This is the F2 phenotypic ratio, right, which you get 9331. Any problems? Now, is this the expected ratio or is this the observed ratio?
Expected.
Right, this is the expected ratio. Okay, now in the question you're going to see sometimes they will give you the observed numbers. So for example, they will tell you that, oh, the F2 generation, there are like 90 plants to 30 to 30 to 1. You have to relate it to the expected. Okay, you see some questions later. So in this case, there's no observed number, so you do not need to relate it. Okay, but in other questions, you have to relate if you see. Any other questions? Let me see whether I cover all the common mistakes. Oh, typo here also. Okay, Ken? Ken? No problems, huh? So, based on your 9331 ratio, okay, you remember you need to memorize your Mendelian ratio. 9331 is when you cross two double heterozygotes together. Alright, so that's how you do it. Any questions, huh? So, please note where are the marks allocated for the F2 part. Right, so again, 1 mark for the gametes from the F1. Can you all write down in your own answer where are the marks? 1 mark for the gametes. 1 mark for the Punnett square showing the F2 genotype and phenotype. 1 mark for the F2 phenotypic ratio. Okay, I'm going to zoom out. Can somebody take a photo? This is question 4. Oh yeah, I forgot the symbols. Still have the symbols. Have you all written down the marks scheme? You have to know where the marks are. Okay, yeah, the symbol also. Listen, thanks. Yeah, why? Okay, good.
Don't write symbols.
Huh? Don't write symbols. Cannot, because you have to let the examiner know that you understand which is the dominant value, even though the alphabets are given in the question. Okay, anyway, can we move on to the next part? Okay, now this part here is showing you another cross. So you have another cross which is white flower long stem with unknown genotype cross with pink flower short stem plant. Alright, now can you figure out the parents? The first one, the white flower long stem with unknown genotype, what is the possibility?
A dominant.
Okay, big A,
And big B.
dash, good. Okay, this is a scalar. If you don't know, you gotta put a dash. And then big B, and then dash. Okay, because white flower long stem. So this is unknown. And you cross with pink flower short stem. So what is the genotype?
Small a,
Small b. So what type of cross is this? Huh? Test cross, good. It's a test cross because you don't know what is this plant's genotype. It's showing the dominant trait, and you want to cross it with homozygous recessive plant. So that is a test cross. So over here, after you cross this, you get these 4 phenotypes with these numbers. Now what do you think the numbers are there for? Ratio, very good. So what is the approximate ratio? 1, 1, 1, 1. So what do you think is the parent? The unknown parent?
Heterozygous.
Yes, remember you have to memorize the ratios, right? So if it is a double heterozygote, okay, crossed with this, it will give you a 1, 1, 1, 1 ratio. Okay, so the question now is how do I explain it? So when they say explain why the results of the cross differs from the cross described in A. Now cross described in A, which cross are they talking about? Are they talking about the parental cross or the F1 cell?
F1
Parental.
Hello, they are talking about the parental cross. Because you are crossing white long with pink short. So they are talking about the parental cross. Now in the parental cross, you get a different result. In the parental cross, you get all white long. Right? So why is that so? How come the results differ? Even though the phenotypes that you're crossing is the same. Yes, okay, so in this parent, you have 4 possible gametes. But over here, the first parent, only 1 possible gametes, right? Because the original parental cross is this, cross with this. Do you see the difference? Right, so this will lead to all white long. Okay, so anyway, so based on all of this, what should you write in your answer? What do you think is the first point? Yes, you have to talk about, you have to name this cross that is being done. Okay, it's a test cross. Okay, now, what then do you think is the second point? Yes, okay, what is being crossed? So in this case, the unknown parent that is unknown genotype, what? The white flowered long stem plant with the unknown genotype is a heterozygous at both gene loci in question. So basically he is this rather than this. Okay, so it is a test cross. Number 2, you have to tell me what is the unknown genotype. So the white flower long stem plant with the unknown genotype is actually heterozygous at both gene loci. So it is big A, small a, big B, big small b rather than big A, big A, big B, big B, which is in the previous question. Alright, so therefore, what is the third point? Gametes. Okay, therefore the gametes formed, okay, from the parent will be 4 different types. How come there's 4 different types? What is the reason for the 4 different types? Yes, okay, instead of, only this, due to law of independent okay, it's only possible because the 2 genes are located on different chromosomes. Yeah? Of course, as long as it's a gamete, it needs to be circled. Otherwise, we don't know it's a gamete. Okay, so basically, it is a double heterozygote, or you can, I think it's better to say it's a heterozygous at both gene loci, right? There's 4 different types of gametes formed due to independent assortment and segregation, right? You have to mention also the 2 genes are located on different chromosomes. Okay, and then what do you think is the last point?
Ratio.
Yes, ratio. So therefore, when crossed with a homozygous recessive plant, which is pink flowered short stem plant, okay, the offspring conform to 1:1:1:1 ratio. Okay, Ken? Somebody take a photo. Ah, yeah. This is for me. Okay, so any questions? Is it doable? It's okay, right? Okay. Can I move on? Okay, so the next question number 5 is a question that we are gonna learn something new. Okay, so can I continue? Any problems with the recording? Is it still recording? Okay. Alright, moving on. Okay, now this question we are looking at a cross between a round-leaf tall plant and a round-leaf dwarf plant. Right, the offspring ratio that you get is all of these, and they tell you already the 2 gene locus. So the first gene locus is for what? The first gene locus, gene R locus, is for leaf shape. Okay, so that is the trait. The second gene locus, gene T, is for
Lines.
okay, plant height. Leaf height?
Okay.
Plant height, okay? So they already tell you what is dominant and what is recessive, okay, for each gene locus. Now, what is the numbers for?
That's all.
Ratio, but okay, if you want to look at the ratio, it's very weird. You have never seen this ratio before. It is around like this. 3:3:1:1. And because of this, then how to do this question? Very good. You need to separate the gene locus. Was it covered during lecture? Okay, so this is a skill that you need to learn. Sometimes you need to separate the gene locus. Very important skill. It's going to come back again and again in this whole tutorial. Okay, so what does separate the gene locus mean? Okay, right now it is difficult because you're looking at 2 different characteristics, and the offspring you're mixing up the characteristics together, right? So you're unable to tell what is the parent actually. But if you were to separate out the gene locus, it is easier to tell what is the parental genotype. Okay, so how do you separate? So the numbers, what you can do is you can group up. Okay, so firstly, group up the round versus the oval. Okay, round and oval, so you first group it up. Alright, so round is to oval. Let's look at the gene R for leaf shape first. Right, so if you look at the first gene loci, round is to oval. It will be around, what is the ratio? The ratio is about 3:1. Now, based on the ratio, of course, based on the ratio, what do you expect the parents to be? Heterozygote cross with? Good. So heterozygote parent cross with heterozygote parent to give you this ratio 3:1. Okay, so based on that, you already can fix. Okay. Oh yeah, I forgot to say. Now the parent, right? Can we look at the parent first? What is the likelihood of the parent? Okay, round leaf tall plant. So this parent will have to be big R with dash and then big T dash. The other one, other parent, big R dash, small t, small t. So right now we already found out that for the first gene locus, it is two heterozygotes crossed together. Okay, so it has to big R, small r, big R, small r. Okay? So right now we have to deal with the second gene locus. So the second gene locus, I'm going to look at the plant height gene locus. So I'm going to group up the tall plants with the tall plants and the dwarf plants with the dwarf plants. So what is the new ratio?
1.
Okay, so the plant height 1, it will be, let me see, plant height, it will be tall is to, no, not short, dwarf. Dwarf. Okay, it will be 121 plus 42 is to 124 plus 37. It will be around 1:1. Okay, based on that, what is the parents that are crossed? Cross with homozygous recessive. So these are the parents. So based on that, you go back to the unknown parent. You will then know that it has to be, this is a small t. So based on that, your answer is A. Okay, I'm gonna just zoom out a bit, then you can take a photo. Is it okay? So this is a skill that you need to have. Sometimes you have to consider the gene locus separately. Alright? Okay, Ken. Take a photo already for your beloved friends at home. Okay, so the next question, question 6. Anybody still need history? Okay, now the question number 6, the Gardner P question. So basically, this is also a dihybrid question. There are two gene locus, I'm sure you know already. First gene locus is seed color. Second gene locus is responsible for seed texture. Right, so these are the two gene locus. Now then you have a parental phenotype plus progeny phenotype. So the progeny phenotype, let's figure out first. So the first question is actually very important, right? How do you know which are the dominant alleles? If you look at these 4 crosses with these 4 offspring ratios, how do you know which are the dominant alleles? So over here, if you look at seed color, right, do you expect the allele for green to be dominant or the allele for yellow?
Yellow.
Yellow. How do you know? Which cross number do you look at? 4th one. Good. Okay, if by cross 4, you should be able to tell that the yellow seed color is dominant. How about the seed texture gene locus? It has to be the round allele. I mean the allele for roundness, right? How do you know? Also cross 4. So how do you explain your question A? Now state which allele is dominant and explain your answer. So this explanation part, you have to cite, cite data. Of course, you have to choose which data to cite. So first mark, you're going to say the allele for yellow seed and allele for round seed are dominant. Second mark, so what are you how are you gonna explain? You're gonna say in cross 4, cross 4, what happens in cross 4? Yes? Yes, when the, okay, so wait, when you cross this parent with this parent, yes? Yes, 100% of the offspring is only yellow and brown. So what does this show?
That is fully expressed.
Oh, it's fully expressed. On the other hand, you can say that the green and the wrinkle has to be recessive. Okay, so it doesn't appear in the F1 generation. Okay, so I mean the explanation is a bit long. Let me just show you the answer. Okay, so the idea that in cross 4 only progeny with yellow and round seeds are produced. Okay, so the idea that in your offspring I only see yellow and brown, even though my parents there's greenness and there's wrinkledness. Okay, so based on that, it means that the green and the wrinkled alleles are recessive. So i.e. the trait has disappeared, right? Even though the parent has greenness and wrinkledness, when you cross the offspring, somehow it disappears. Okay, so because of that, it has to be recessive, the two alleles. You all get cross 4? I hope so. Okay, now the next question we have to use suitable symbols. So we have to assign symbols. Alright, so just because it's just easier to remember, we will use the symbols related to the phenotype. Okay, so for yellowness, okay, we are gonna use big Y. Okay, let me just zoom out a bit. Okay, so over here, let me see. For greenness, we will put yellow. This one is green. And then brown and wrinkled. Okay, so just for ease of remembering, we are going to use these symbols. And these symbols, you have to write it in part 2. Okay, so let's look at the question. And anybody, have you all taken a photo of this? Curve. Please take. Okay, okay, then I'm gonna move on. Can okay, now the next part. Now over here, let's say we have the symbols already. Okay, say the genotypes of the parents. We will use Y and R for the symbols. Okay. Now, how do you know what is the parents in the form process? Okay, so let's take a look at the offspring first. The first offspring that is yellow and brown, it is likely to be big Y, and then followed by the first one, yellow and brown. Big Y with small y, you don't know yet.
Dash.
So dash, and then
Big R.
big R,
Dash.
dash, okay? And the yellow wrinkle is like this, right? The green brown is like this, and the green wrinkle is like this, right? So that is your offspring phenotype and genotype. Okay, now we have to look at the parents. So how do I get a parent? By the way, the offspring ratio for cross 1 is quite clear. What is the ratio? 1, 1, 1, 1. right? So the ratio is 1, 1, 1, 1. And the parents, let's try and figure out the parents. The first parent being yellow and brown, it is likely to be big Y dash, big R dash, and then green wrinkle is small y, small y, small r. So based on the phenotype ratio of the offspring, 1, 1, 1, 1, the parent has to be heterozygous at both gene locus. So you can get cross 1 super easily if you memorize the ratio. Now cross 2 is the most difficult one. I'll skip cross 2 first. We look at cross 3 first. Now cross 3, the first parent is yellow and brown. So I'm just going to write this first. The second parent is yellow and wrinkled. How do I know what is the dash? Okay, if I look at the offspring ratio cross 3, what is the offspring ratio? It is like 3, 1, 1, which is very odd. So there is no ratio, I know there's 3, 3, 1, 1. So what should I do? Separate. Yes, you have to separate the gene locus for this question. So how do you separate the gene locus? So let's put another column at the side here. And we write offspring. Okay, so if you look at the offspring for cross number 3, cross number 3, if you look at the offspring, yellow is to green. It will be what? What is to what?
3 to 1.
It's 470. If you add all together, it's to 161. So it is 3 to 1. I hope you know what I'm talking about. Yellow and green. So basically, yellow, all the yellow offspring, you're gonna group them together and add them up, right? And then the green one, 75 plus 86, add together. And then you get a ratio 3 is to 1. Alright, now the next one you're gonna do round is to wrinkle. What is the offspring ratio for round and wrinkle? Okay, if you look at round and wrinkle, let me just erase this. Roundness is here, this one plus this one. And then the wrinkle is this one plus this one. So if you add up, it will be 1 is to 1. Am I doing it right? Okay, so anyway, it will be 298 is to 333. You add up, and it will be equals to 1 is to 1. Okay, so if you separate the gene locus, you can then find out what is the genotype of the parent. How do you get an offspring ratio of 3 is to 1? If you cross what with what? 2 heterozygous. Okay, so it is like this. Right, and the second gene locus about the roundness versus the wrinkledness part. How do you get 1 is to 1? Heterozygote with homozygous recessive. Okay, so based on that, that is your cross 3 genotype of parents. Next one, let's look at cross 4. Cross 4, we have green, brown, and then we have yellow, wrinkle. So this is what we can gather so far based on the parents, right? To be able to find out the parents, let's look at the offspring. Is it any ratio that we are familiar with? Not really. Okay, we have all yellow and brown. So there's no ratio that we know. So you should also, you can, if you want to separate gene locus, you can. But anyway, how do you get the parent genotype for the first one? In order to get all yellow, your offspring all yellow and all round, okay, what do you think is the offspring genotype? It has to be heterozygous, right? Because the parents are different. Okay, so all round, it has to be like this. So it means that the parents have to be homozygous. So it has to be like this with like this. Can? Okay, so the most difficult one is cross 2. Okay, so we look at cross 2 next. Cross 2, you have yellow, brown, and yellow, brown. So this is the genotype that you expect. So for this question, if you look at the ratio, it's not really any ratio that you can figure out is 3 is to, I don't know, 0 and then 1 and then 0. So based on that, you of course you have to separate gene locus. Okay, so I'm going to erase everything, I'm very messy. So if I were to separate the gene locus, so we look at cross 2. Okay, so we look at yellow and green first. Alright, so what is the ratio for yellow is to green?
3 to 1.
Yellow is to green is 176 is to 60 is 3 is to 1. Then the next one we look at round and wrinkle. Round and wrinkle, if you group them together, round and wrinkle. What's the ratio?
All round.
It is 236 is to 0 there, which is all round. Okay, so based on that, what is the parental genotype? How do you get this? Okay, so let's take a look at the yellow is to green part first. How do you get a 3 is to 1 ratio? Okay, it has to be 2 heterozygotes crossed together. Then how do you get all round? Yes, it could be one that is homozygous crossed with another what? Homozygous. Is that all? Yes, it could also be this. Okay, so this question is difficult because this, it can be either. Right? It will still give you the results of all round. Okay? Yes, you need to write bold. Yeah, you need to write bold. Okay? Alright, now the next question. Take a photo already. Thanks. Okay, last question for today, and then 8 and 9 will be covered during your lecture slot on Thursday. Alright, so I move on. Now question number 7, okay, you have 2 heterozygous pea plants grown from round yellow seed. Okay, and then these plants were crossed and then the seeds produced were recorded. So over here, right, they say that the plants were heterozygous. So let me just put a symbol first. Okay, this cross with this, alright, give you this. Right, and you expect a 9331 ratio, they said. Okay, so it should be 9331 ratio, right? Now the first question is a Cambridge question. I mean, this whole question is Cambridge question, but this question is asked quite frequently. Okay, so what is the question asking? Do you understand the question? Okay, they are asking you, why is it that the phenotype of the seed is not exactly 9331? If you want it to be exactly, it will mean that it has to be 270, 90, 30, 90, 90, 30. Okay, this is what I mean by exact 9331, right? But this is an approximate 9331. So the question is, how come it's approximately and not exactly? Why? What is the first answer? Random fertilization, yes. Okay, so you can say due to chance event. Okay, random fertilization. Okay, so if you look at, for example, if you look at the Punnett square, that's my Punnett square. If you look at the Punnett square, I cannot predict which gamete formed from either parent is gonna fuse to form what offspring. Okay, so that is random fertilization is a chance event. Alright, and if you don't understand what is a chance event, the best example is tossing a coin. Okay, so which is mentioned during the lecture. So if you have a coin and you have heads and tails, right, I toss the coin 10 times. Okay, what do you expect the number of times to get heads and what is the number of times I expect to get tails? You expect it to be 5 times head and 5 times tail. But even if the coin is unbiased, that means it's a fair coin, it's not like somebody make the coin differently. Right? Even if the coin is unbiased, I cannot get exactly 5. Sometimes I get 6 and 4. So this is because it's a chance event. The more you want it to approximate the ratio, what do you have to do for it to go closer to the expected ratio? Yes, you got to increase sample size. Okay, that's why when scientists do research, they have to have a large population to be more valid. So you can also talk about sample size. So you can say sample size need to be larger to get closer to the expected ratio. Any other possible explanations? Yes, some seeds died, exactly. Okay, you can talk about adverse environmental conditions. Okay, some seeds might have died. Anything else? Mutation, no lah. We are talking about like in the natural environment without things like mutation and everything. Now there's one that is quite lame, but Cambridge accepted it. It's called incorrect counting. Alright, which is possible. You know why? Because it has to be a human that's doing all this counting, right? And if you count something so large, it could be that you counted wrongly. So that's what they said. Anyway, the last part of the question, can I just show you the answer? It's not difficult. Can you just check and that's it? Okay, and then you can do your cleanup. Okay, so for the cross, can you check your own answers? Oh, by the way, do you have to clean up or do you have a lesson here? Okay, can you make sure you write down where the marks are allocated? Okay, and when we continue the next time, I see you. Bio rap. Please take photos. Our lessons are not affected by the Friday thing. Previous answer. Anybody still needs these? Okay, Noah. This one? Alright, please go and clean up. Yes? Yes, we uploaded some stuff on SLS in preparation for home-based learning. Okay, we will give you instructions, don't need to do yet. Okay? Okay, please clean your desk.